Thank you very much for the introduction and for organising this really interesting session. And yeah, today I'll be telling you a bit about how we're incorporating nanopore technology to bridge genomic research with conservation of endangered sea turtles. So it's really important for conservation programs to integrate knowledge on how temperature affects climate vulnerable taxa in order to guide suitable interventions as climate change progresses. And one group that's especially at risk is over 400 species of reptiles and fish with a temperature-dependent sex determination system, or TSD for short. And so this is where instead of having sex chromosomes, their sex is determined by temperature during a thermosensitive window of egg incubation, with the three main reptilian systems shown here. So these are described by a 1-to-1 sex ratio at the pivotal temperature and a transitional range of temperatures where both sexes are produced, shown here in orange. So, of course, with global warming, this could be bad news as these species face heavily skewed sex ratios and ultimately demographic collapse if they can't adapt quickly enough to rising temperatures. And sea turtles are one group of TSD species of special conservation concern, as they're a set of 7 already endangered species facing many compounding threats, such as bycatch, pollution, poaching, and coastal development, to name a few. And they're also charismatic or flagship species, which means they're some of the most heavily managed species in the world as wild marine vertebrates. And so this means, as we're super involved already, we need to make sure our management strategies are appropriate and science-driven, including in relation to this additional risk of temperature sex determination and skewed sex ratios, as their TSD system produces males at cooler temperatures and females at warmer temperatures. And of course, so many people are concerned about this, such as studies projecting complete feminization by the end of the century under future climate scenarios. And worryingly, these types of effects are already starting to be seen, such as in this Great Barrier Reef population where over 99% of young green turtles were already recorded to be female in 2018. However, a major flaw in our field is that most studies of wild sex ratios, including this one here, didn't actually measure the nest sex ratios, but rather used sand or nest temperature as a proxy for sex ratio. And this was using a mathematical relationship between temperature and sex that was calibrated with TSD parameters obtained about 20 years ago with a small set of constant temperature lab experiments. So obviously, wide use of this proxy method could be problematic, as firstly, it doesn't capture the impact of complex field variables on the temperature-sex relationship, and also the TSD parameters are unlikely to represent different populations across the globe, which are locally adapted to diverse thermal niches. And the main driver of this gap is that there's still currently no way to tell the sex of newly hatched sea turtles without sacrificing them. And obviously you can't do this as it's highly restricted, they're endangered and protected species. And the current only method for non-lethal sexing is to rear the hatchlings for 3 to 6 months until they're large enough for laparoscopy, which you can do to surgically examine the gonads. However, this is also unfeasible at scale, as it's difficult and expensive, and it has unknown biological effects on key life history traits, such as imprinting and therefore later survival. So, without a method to easily assess sex ratios empirically in the wild, we also don't understand how climate change is affecting them and therefore what interventions are actually required. And so we need a scalable and non-lethal method for sexing these hatchlings as a monitoring solution, which we think could be provided by epigenetic biomarkers, specifically focusing on DNA methylation here, which is the addition of methyl tags to cytosine residues in the DNA to regulate gene expression. So yeah, certain properties make methylation a really promising biomarker to use, as it can firstly represent individual level regulation of both developmentally innate and environmentally inducible traits. It can stably maintain regulatory information, often across life stages, and also on the DNA molecule itself, so you don't need special storage conditions. And it's also amenable to study with a wide range of established pipelines for non-model species. Okay, so we're identifying these biomarkers from circulatory blood, as this tissue type is well suited to monitoring protected wildlife, as you can obtain it via minimally invasive sampling. And indeed, these previous studies did find methylation sex differences from blood, including in humans and promisingly in alligators, which are another TSD species. So to tackle our aim, our key study site is in Cabo Verde, which is just off the coast of West Africa, and hosts one of the largest loggerhead sea turtle nesting populations in the world. And here we set up a project with 3 main phases, starting with step 1, which was to generate a high-quality reference genome for our loggerhead population, as this is a prerequisite for genomic studies, as I'm sure you all know. So for this, we collected blood from a nesting adult female and extracted high molecular weight DNA for sequencing, which we chose Nanopore Technologies for, as it allowed us to generate long reads for de novo assembly, while simultaneously characterizing methylation modifications, which is important to us, and I'll go further into later in this talk. So this assembly pipeline gave us a chromosome-level reference assembly with 98.9% of contigs scaffolded into 28 chromosomes, and a BUSCO completeness of 97.1%, and over 29,000 annotated genes. So we were happy with this, as it allowed us to move on to step 2, the meat of this project, which was to identify methylation-based biomarkers of sex from hatchling blood samples. And this was only possible through our collaborators at Florida Atlantic University, as they can obtain permits to retain some hatchlings for sex verification via laparoscopy, which is highly banned in Cabo Verde. So they took eggs from these wild females and individually incubated them at 3 different temperatures to induce different sex development in artificial nest boxes. And then upon hatching naturally, they took a small blood sample from each hatchling and then raised the hatchlings until they were large enough for laparoscopy to identify their sex. And then we extracted DNA for whole genome bisulfite sequencing, as this was the classic gold standard method at the time for base resolution methylation characterization, as this allowed us to identify differentially methylated sites, or DMS, between the sexes that we could use as a set of biomarkers. And so from our epigenome-wide scan, we identified 777 sex-specific DMS, which, as you can see, strongly clustered the hatchlings by sex in this PCA plot. And these were distributed across all chromosomes, with over 400 associated to diverse genes, including promising standalone candidates such as AMDHD2, which is a spermatogenesis gene that was hypermethylated in female hatchlings, and also found to be hypermethylated in female children in blood and saliva samples. And so this could be interesting, as it hints at possible cross-species and tissue universality at this site to look into in the future. So after finding a set of sex markers from the Florida lab experiment, we next applied them to assign hatchling sex from a larger scale Cabo Verde field experiment. And this involved teaming up with the local NGO Project Biodiversity to relocate the clutches of nesting females to their in situ hatchery on a nesting beach here, where we then split the clutches randomly in half and buried them at 2 different depths to induce different incubation temperatures in otherwise field conditions. And we repeated this over 3 time points over the warming nesting season to give 6 time point treatment combinations with 9 to 10 clutches per time point, from which we assigned sex based on our methylation biomarkers for these hatchlings. So this graph just shows that mean nest temperature was indeed different between each time point treatment combination. Then here are the sex ratios predicted from our methylation biomarkers, with the proportion of females on the y-axis. in contrast to modeled sex ratios using the traditional proxy-based methods, which overestimated female production by an average of 52.4%. And so this discrepancy really highlights the importance of shifting the field towards empirical sex ratio assessments on a population-specific basis, which moves us nicely onto step 3 of this project, which is currently in progress. And this involves developing a method for large-scale deployment of our sex biomarkers in a conservation context. And for this, we needed an accurate, scalable, and portable sequencing solution, which Nanopore Technologies uniquely met all of these criteria for us, specifically focusing on the P2 Solo for our research needs, as it's very small and portable, at just about this size, but it has a higher throughput than the MinION model, as you can run 2 PromethION flow cells simultaneously. And I'm excited to share that last year we successfully trialed nanopore sequencing in the field in Cabo Verde. So we went from hatchling sampling on the beach to over 140 gigabases of data for 8 hatchling genomes in about 3 days. And a key part of this success was the rapid barcoding kit, as the thermal stability and the simple protocols made it possible for us to do all of the lab work in our hot hotel room with just a single suitcase of equipment. And here are just some reports from the sequencing run, just to emphasize that high-quality data was indeed possible in these field settings. So this is just the cumulative sequencing output with the passed bases in green for one of the flow cells, and the base calling quality score, which remained above Q10 for the entire run. Oh, missing a slide. Oh well. And yet, crucially for us and our research goals, nanopore devices can perform methylation calling simultaneously as genome sequencing. And so we don't have the data yet analyzed for the field work. However, this density plot on the right here is the global methylation profile per gene feature type from our reference genome nanopore reads. And these were highly consistent with WGBS data from the same population with an R-squared of about 0.92. And so this supported that nanopore methylation calls are indeed capturing expected patterns for this non-model species. But obviously, it's been well benchmarked with the same samples in other species too. So our next step moving forward to maximize scalability is to use adaptive sampling to target the sex biomarker regions. And this works by smartly ejecting reads that aren't target sequences from sequencing nanopores in real time using a supplied bed file. As we want to roll this out as a portable sexing tool for our NGO partners in Cabo Verde to use to ultimately guide hatchery management decisions in real time, such as nest shading location and, throughout the nesting season. And of course, perform large-scale sex ratio assessments on a global scale through our network of international collaborators who are now also looking into getting a P2 Solo. So just to conclude, I hope I've convinced you that methylation biomarkers can be a powerful tool for wildlife surveillance. Today focusing on sex markers for sea turtles as they're threatened by extremely skewed sex ratios as climate change progresses. And Nanopore Technologies is uniquely suited to drive our research forward as it offers a portable solution for on-site methylation sequencing that none of the other available models can do currently. And this provides a special case where genomic tools can directly influence management decisions for an endangered species in real time. And with that, I'd like to thank everyone involved in this huge collaboration and happy to take any questions.